Hi guys, welcome to class. So, really, you see, early diagnosis of bacterial infection is very key. It's very, very, very key. That's the reason why I do say, when you have used an antibiotics, it doesn't work, do not move over to another antibiotic. There's nothing like this antibiotic is stronger than this one. That antibiotic has a spectrum of activity. That is, it only caters for a certain class of bacteria. If it's not going to work, it means that the antibiotic you are using, the spectrum of activity does not include what is causing your infection. So don't go antibiotic shopping. Why? Because early diagnosis of bacterial infection is very key. Some bacterial infection starts very slow because they have the ability to reproduce. So they start minor, you don't detect it on time, you don't nip it in the bud, it could progress. There are certain bacteria which you might not know you are infected with that have the ability to cause the blood-brain barrier and can lead to conditions such as encephalitis or meningitis, inflammation of the membranous coverings of the brain. So, early diagnosis is one of the ways of preventing, not should we say prevention now, control. What we looked at the other time is more of prevention, it's satisfied prevention. But control is one of the ways of trying to control it. Early diagnosis, so you get nip in the bud. It's also good because it prevents further spread. Because if you identify it, you put measures, preventive measures that prevent further spread. And also helps for effective management of that infection. Also, another way of controlling bacterial infection that in the hospitals, having good working medical equipment are very necessary because they will be required for diagnosis. When we are making diagnosis, you need to use good medical instrument because particularly in bacterial infection, you need to look at slides under the microscope. So, good microscope, that's the reason why it's important that the hospitals are well equipped so that quick diagnosis can be made and treatment can be started as soon as possible. As well as clean syringes because the idea is infection. Because there are hospitals where the same medical instrument that was used on one patient will be used for the other patient. Even some hospitals can't run the process of sterilizing equipment, even surgical equipment effectively. These are things that can bring about spread of infections. So if you're talking about controlling infections, good working medical equipment, use of clean syringe, not sharing medical equipment, and also on the part of the victim or the part of the patient, I won't say victim, early diagnosis is necessary. So go see a doctor for early diagnosis. Now, something we want to talk about, and that is measuring the growth of microorganisms. Now, in this case, our concern is bacteria. So now, there are actually two major methods. In this video, we'll look at the first major method, the method of determining bacterial growth. Now, the first method is the turbidity test. Yeah, turbidity test. Now, it makes use of a equipment called the spectrometer. You see, actually, generally, we measure bacteria unit of micrometer. So when you have the more the micrometer, it's more likely to say there's more bacteria. Density of bacteria is much higher. Because the average bacterium, that is the average bacterial cell, is just about 1 micrometer. So the more the micrometers, it's more like the more the density of the bacteria. Now you see, if you take a pencil drop, a drop of a pencil contains millions of bacteria. They are so very, very tiny. They are so very, very, very tiny. Though you can still see them with light microscope, you can see them with light microscope. They are not as tiny as viruses that you need electron microscope to see. Now in spectrometer, you are just going to be playing around with the principle of turbidity because you have a petri dish. So now basically, when something is turbid, light penetration is low. You understand? But when something is less turbid, light penetrates more. So that's just what the spectrometer is trying to measure. Because when you see because measure a culture bacteria, most bacteria, there's some bacteria that you culture them in a tube, in a nutrient agar. Most bacteria, you culture them on a petri dish. So when you look at the petri dish, for instance, you start culturing the bacteria. As we go further, we'll still talk about culture. So culturing methods. So you start culturing the bacteria, you will see that the bacteria will be multiplying, is reproducing, Now, when the light intensity drops, so you look at the progress of the drop of light intensity, that is light penetration. Now, when the light penetration drops, it's indicative that the microorganism, that is the bacteria, has increased sufficiently in size, and is also a measure of turbidity. So the higher the turbidity, the lesser the penetration of the light, and the more the number of the bacteria. So that's how you measure. So because if there are more bacteria inside that petri dish, there will be more turbidity. With more turbidity, there will be less penetration of light, less light intensity. That's a very, very good, quick method. Makes sense, right? Yeah, turbidity test. That's how we measure growth of bacteria in laboratory. We're looking at the other method, which is called the nutrient agar inoculation method, in the next class. See you next one.